Right. So welcome to the lecture, everyone. So my name is Hui Chu. Now I am a postdoc research fellow in the Department of Environmental Health and in the Mahalingaya Lab, and I will be your moderator for today's lecture. So a little bit introduction for this lecture series. So the goal for this lecture series is to expand our inclusion of women's health outcomes into environmental and more broadly public health research. So if you're interested in our research series for the rest of the semester and in the future, please stay tuned for the invitation on our lectures in your emails. So a couple of rules before we start the lecture. Please note that this lecture is being recorded. And during the lecture, you may put your videos on or leave it off. It's up to you. And as for questions, we will have a Q&A session at the end of this lecture. So if you have any questions during the lecture, you can enter them in the chat box and we'll take care of them at the end. Or you can hold your questions until the end of the lecture, and then you will have the opportunity to use the raise hand function and ask them yourself. So please let me introduce today's lecturers, Dr. Emery Yukish. So Dr. Yukish is a perinatal epidemiologist whose research spans the perinatal period, including preconception, pregnancy, and pregnancy outcomes and child health. She's interested in early pregnancy, particularly implantation and early placental development, and the relevance of these events for pregnancy health and child health. So her current research focuses on the role of vitamin D in reproduction and has published her first papers describing the link between vitamin D and menstrual cycle function in a community-based samples of women. Dr. Yukish received a Bachelor's of Science from the University of Notre Dame and a MSPH from Emory University, and then finished a PhD in epidemiology from the University of South Carolina at Chapel Hill. And later, she completed a doctoral fellowship, postdoc fellowship at NIEHS and served as assistant professor at the Yale School of Public Health before joining NIEHS as a principal investigator. So today, she will be presenting her amazing work on vitamin D and reproduction. And without further ado, let's welcome Dr. Yukish.
Thank you, Ichu. Is it okay if I share my screen?
Yes, please go ahead.
All right. I think I want that one. Does that look good? Excellent. Thank you so much for that introduction. I'm really glad to be here. I appreciate the invitation. I'm happy to describe my work on vitamin D and reproduction, and I'm going to move through some observational studies today into a clinical trial that we just started in February. I just want to point out really quickly that my research is targeted towards understanding fertility and reproductive success. And there are two primary research areas that I work in. Vitamin D is obviously one of them, but I'm also interested in air pollution and climate change. But for today, I'm just going to talk about vitamin D, but I also have interest in those other research areas. So in my presentation today, I'll provide background on fertility and vitamin D, including their public health significance. Then I'll describe my research within vitamin D and several reproductive endpoints, including menstrual cycles, conception, early pregnancy growth, and pregnancy loss. And I think this leads naturally into the description of a clinical trial of vitamin D that we just recently started. And then finally, I'll talk about some in vitro and mouse studies that we're also working on. And there's some preliminary data from both of these research projects that supports the hypotheses we're investigating in the trial. So I think they fit in nicely with the description of the trial. Okay. So in the most recent, I'm going to move this, sorry. In the most recent iteration of the National Survey of Family Growth, 12% of reproductive age women reported that it was difficult or impossible to conceive a pregnancy. And for each person reporting that they have difficulties conceiving, there may be a partner sharing that experience. So that would increase then the number of affected persons. Moreover, this is the proportion of women currently experiencing fertility problems. The proportion experiencing fertility problems in their lifetime would, of course, be higher. Fertility treatments that help people conceive are expensive. For example, in vitro fertilization can cost $15,000 to $20,000 per cycle, and multiple cycles can be required to achieve a live birth. Additionally, there are known disparities in fertility treatment utilization. Utilization is lowest for people of Black race or Hispanic ethnicity. Some states require that fertility treatment be covered by insurance companies, and these insurance mandates increase utilization rates across all racial ethnic groups, which indicates that cost is indeed a barrier. However, the disparity across racial ethnic groups remains. Thus, it is important to identify the causes of subfertility to reduce its burden, but it may be especially important for populations lacking equal access to the remedy for subfertility, which is fertility treatment. So vitamin D is known as the sunlight vitamin, and that's because it's synthesized in the skin in response to ultraviolet B radiation. These cholesterol precursors in the skin are converted to vitamin D3. Vitamin D can also come from our diet. It's found in fatty fish and in fungi. The form of vitamin D in mushrooms and other fungi is vitamin D2 as opposed to vitamin D3. However, in humans, vitamin D3 is much more common. So for the purposes of this presentation, I'm just going to refer to both forms or the total as vitamin D. Vitamin D from the diet is absorbed in the intestine and it circulates in the blood. Similarly, vitamin D from the skin is carried in the bloodstream. And vitamin D circulates to the liver where it's converted to 25-hydroxy vitamin D, which is also called 25-OHD for short, which is often how I refer to it in this presentation today. 25-OHD is the circulating biomarker of vitamin D status, and it's what is used clinically to define vitamin D deficiency. There's some disagreement about that, but one cutoff that could be used to define deficiency is 20 nanograms per mil or less than 20 nanograms per mil. And then sufficiency could be defined as the converse, greater than or equal to 20 nanograms per mil. 25-OHD can be further metabolized to 125-dihydroxy vitamin D, or as an abbreviation, 125-OH2D. This is the active form of vitamin D, and it binds with the highest affinity to the vitamin D receptor. This is the ligand for the VDR. The conversion of 25-OHD to 125-OH2D is catalyzed by the enzyme 1-alpha hydroxylase, or CYP27B1. The kidney is the most well-known site for the conversion of 25-OHD to its active form. However, the discovery of 1-alpha hydroxylase in numerous other tissues suggests that this conversion occurs elsewhere, including the ovary, the uterus, the brain, and the placenta. This has led to the hypothesis that vitamin D may be relevant for numerous biological processes, including reproduction. The figure here shows the distribution of 25-OHD levels in the United States collected through NHANES. This is the most recent data from NHANES. The mean 25-OHD level across all reproductive age people is 25 nanograms per mil, which is lower than all other age groups. The prevalence of vitamin D deficiency varies by race and ethnicity, with a prevalence of 75% among Black people, 45% among Hispanic people, and 14% among white people. So to bring all of this together, vitamin D and fertility, the observational studies I've done were motivated by a handful of animal studies that are based on 3 animal models. The first is the vitamin D 1-alpha hydroxylase knockout mouse. I mentioned that 1-alpha hydroxylase is the enzyme that converts 25-OHD to its active form. There's also the vitamin D receptor null mouse model, where the vitamin D receptor is knocked out. And the dietary restriction model, where vitamin D deficient diet is fed to mice and rats. So somewhat surprisingly, all three of these models suggest reproductive effects of vitamin D. In these deficiency models, there was decreased follicle development, decreased ovulation, decreased corporal lutea, increased uterine hypoplasia, and decreased fertility. So based on those animal studies, I completed a series of studies of vitamin D across a spectrum of reproductive endpoints, from menstrual cycles to conception to early growth to pregnancy loss. My presentation today will walk through this spectrum, beginning with the series of observational studies, both published and ongoing. And so my first research question based on those animal studies was, is vitamin D associated with prolonged menstrual cycles or delayed ovulation? We first examined vitamin D in menstrual cycles in two cross-sectional analyses. And we found that lower 25-OHD was associated with increased odds of either prolonged or irregular menstrual cycles. So we wanted to follow up on those data with, because they were based on self-reported cycle length, we wanted to follow up with a prospective study. And I'm going to spend a couple minutes talking about the time to conceive cohort because several of the observational studies we've done have been within this cohort. So our analyses of vitamin D were completed with time to conceive data. Time to conceive is a prospective time to pregnancy cohort of women aged 30 to 44 in North Carolina. And at enrollment, women provide a blood sample and then they're followed for conception or a positive home pregnancy test, which was provided by the study. Their time in the study can be divided into menstrual cycles through their daily and monthly diaries. So they record their menstrual bleeding within these diaries, and then we divide her time in the study into menstrual cycles. As I said, at enrollment, they provide a blood sample and a urine sample, and this blood sample was eventually analyzed for 25-OHD using LC-MS. Also, across their menstrual cycles, they collect information regarding supplement use, medications, and other behaviors like smoking, alcohol, and caffeine intake. The women that conceived in the study were offered an early ultrasound around 7 to 10 weeks of gestation, where they also provided, for a subset, also provided a blood sample and a urine sample. And this again was analyzed with LC-MS. So I mentioned that 25-OHD was measured at enrollment, and then as participants move through time, that measurement may be misclassified. So to address that, we estimated a cycle-specific 25-OHD level. To do that, we built a predictive model of 25-OHD for the menstrual cycle in which blood was drawn. And this was our model, that 25-OHD is equal to season, supplement use, and any other predictive factors, including interactions that are time varying. So only time varying variables are included in this predictive model. Then for each woman, we subtracted the measured 25-OHD from her predicted value, and we called this quantity the woman's residual. For menstrual cycles that occurred outside of the blood draw, we used the model to predict the 25-OHD level in those cycles. Then we add the woman-specific residual to that predicted value to obtain the final cycle-specific 25-OHD level. So we looked at several cycle endpoints. We analyzed continuous menstrual cycle length using a linear mixed model with a random intercept to account for clustering by woman. Menstrual cycle length was natural log transformed, and this analysis was limited to cycles of 22 to 36 days to achieve normality of the residuals. And while long cycles were included from the continuous analysis, they were captured in our dichotomous variable long menstrual cycles. Long cycles were defined as cycles of at least 35 days, corresponding to the clinical definition of oligomenorrhea. And then short cycles were 25 days or less. Both dichotomous variables were compared to cycles of 26 to 34 days. The dichotomous variables were analyzed with marginal models with an exchangeable working correlation matrix and estimated with GEE. In our analysis of cycles, we have 446 women and over 1,200 menstrual cycles. Each woman provided about 1 to 6 cycles, and 90% contributed 1 to 4 cycles. The median cycle length was 29 days. And this distribution shows the results of our 25-OHD assay. So you can see that 5%, there we go. 5% had a level of less than 20 nanograms per mil, and about 31% had a level of less than 30 nanograms per mil. So less than 20 is a definition of deficiency based on IOM cut points, and less than 30 is based on the Endocrine Society guidelines. So we have a small tail here that qualifies as deficient or insufficient. I also want to point out the standard deviation is 10. And you can see the distribution of vitamin D here. Usually, this is sort of unusual. Usually it's a skewed distribution with a lot of observations down on this end. So actually having a broad standard deviation provides us an opportunity to look at relatively high levels of 25-OHD in this population. So these are the results of the 25-OHD and long menstrual cycle analysis. I'm not going to show the continuous or the short cycles because we didn't see any associations with those endpoints, but we did see that a 10 nanogram per mil decrease in 25-OHD was associated with about a 20% higher odds of long cycles. And when we looked at 25-OHD categorically, we saw that the less than 20 group had 3.5 times the odds of long cycles. And then the group in the middle of 20 to 40 nanograms per mil also had elevated odds of long cycles of 1.7. And this is compared to the highest level of 40 nanograms per mil. We did several sensitivity analyses to look at those results, including excluding women with low AMH, which could be an indicator of perimenopause. We tried excluding women with high AMH as an indicator of subclinical or undetected PCOS. We tried using the measured value of 25-OHD instead of our cycle-specific imputed value of 25-OHD. And we also tried accounting for informative cluster size, and our results were unchanged across all of these sensitivity analyses. So then remember that in the animal models, we saw that there was this decreased corpora lutea, delayed reduced ovulation. And so we wondered if 25-OHD in our human study would be associated with longer follicular phases. Now, unfortunately for this endpoint, we had distributed clear blue digital ovulation predictor kits partway through the study. So not all of the women received the ovulation kits, so we have a smaller sample size. We did define ovulation based on the manufacturer instructions 24 hours after a positive test. We used a very strict definition. So if she had a positive test, but she didn't test the day before, we didn't count that. We didn't allow missing days. Then we divided follicular phase length into long and short. And just as a reminder, the follicular phase is this time from the first day of menses up to, but not including ovulation. So this is the follicular phase length. And this over here is the luteal phase length, the number of days after ovulation until the onset of the next menses. So we divided follicular phase length into long and short based on the 90th and 10th percentiles. And also luteal phase length divided as long being 18 days or more and short being 10 days or less. And again, we used our generalized linear model in GEE to estimate the associations between 25-OHD and cycle length as a dichotomous variable across multiple cycles. So the figure shows the predicted probability of a long follicular phase on the y-axis by 25-OHD level on the x-axis. And the risk of a long follicular phase is highest for low levels of 25-OHD, which suggests that lower levels of 25-OHD are associated with delayed ovulation. And since we didn't find any other literature on vitamin D and menstrual cycles or ovulation, we wondered if season as a potential marker of vitamin D status might be associated with either of these endpoints. So we looked at this in 2 different cohorts. First, we looked at the follicular phase length in the North Carolina early pregnancy study. And the early pregnancy study was a preconception cohort of women trying to become pregnant. And in that study, we saw a slight seasonal pattern with follicular phases being about 12% longer in early March versus early September. And this association was stronger in older women, women over age 30, which would compare with our time to conceive study population. Additionally, we used menstrual cycle data from the Trelor study, which was a landmark study of menstrual cycles across the lifespan. It was conducted in Minnesota from 1930 to 1976 and includes almost 1,000 women and over 100,000 cycles. And in those data, cycles in November and December were slightly longer, like 0.4%, than those that occurred in May and June, both overall or stratified by age. So to summarize all of the menstrual cycle studies, lower 25-OHD was associated with higher odds of long menstrual cycles. The lowest frequency of long cycles occurred where 25-OHD was at least 40 nanograms per mil, which is higher than the cutoff for sufficiency by any professional recommendations. And the lower 25-OHD was associated with a higher probability of a long follicular phase. And there was a small seasonal pattern to cycle length and ovulation timing. And in total, our prospective analysis of 25-OHD agrees with the previous cross-sectional studies, and vitamin D may influence menstrual function or ovulation. So far, we have evidence that vitamin D is associated with menstrual cycle length in 2 cross-sectional studies and in the prospective data. So our next research question was whether vitamin D was associated with the probability of conception. And we again used time to conceive data to examine this association. We used the measured value of 25-OHD in this analysis, as the imputed value that I described before wasn't well understood when we had that study reviewed. So it seemed the measured value was actually preferred by the reviewers. We also looked at the probability of conceiving in a given menstrual cycle. This can be estimated with time to pregnancy, the number of menstrual cycles to conception or censoring. If you think of that as K, then 1 over K is a maximum likelihood estimate of fecundability, that probability of conceiving in a given cycle. So our analysis strategy takes into account what we call delayed entry, which means that our participants have cycles of attempt prior to being in our study. A couple doesn't enter the analysis until the cycle they enrolled in the study. Thus, the estimate of fecundability for cycle 1 is only estimated for couples who were enrolled in the study at cycle 1. If a couple didn't enroll until cycle 3 of trying, they would not contribute to our analysis until cycle 3. And just want to point out that in this method of modeling, the hazard ratio is often referred to as a fecundability ratio. And a value of greater than 1 indicates increased fecundability in the exposed group, which would be a good thing, faster time to pregnancy. And then less than 1 indicates decreased fecundability in the exposed group. And this table shows the associations between 25-OHD and fecundability, where fecundability is defined as the probability of conceiving a pregnancy in any given menstrual cycle. In this table, a fecundability ratio of FR of greater than 1 indicates increased fecundability or improved conception. So the first row shows that for a 10 nanogram per mil increase in 25-OHD, there was a 10% increase in fecundability. When examined categorically, a 25-OHD level of less than 20 nanograms per mil was associated with an almost 50% reduction in fecundability, although the confidence interval was wide and consistent with values above and below the null. The reference group is a 25-OHD level of 30 to 40 nanograms per mil. The mid-range of 25-OHD was not associated with fecundability. And then at the higher end of 25-OHD, while the confidence interval is broad and includes the null, a 25-OHD level of 50 nanograms per mil or higher was associated with a 35% increase in fecundability. And since these associations were relatively underpowered or imprecise, we wanted to compare our results with other existing studies. So I just want to show this really quickly before the other, I'm missing a slide. Before the other, I show you the other studies. This is the probability of conception as the probability of taking more than 6 months to conceive across the vitamin D groups. So this is just another way of looking at that table, but it divides the probability into a yes, no, conceiving within 6 months or not. This is important because in this population, 6 months is when it's recommended for women to seek treatment. So the probability of taking longer than 6 months if you're over age 30 is a relevant clinical endpoint. And again, you see that high vitamin D means you're less likely to take at least 6 months to conceive, and low vitamin D means you're more likely to take at least 6 months to conceive. And we did do multiple sensitivity analyses similar to the cycle length analysis, and really the estimates are unchanged across any of these other adjustments. Oh, here we go. So this figure shows the results of the existing 4 studies that have examined 25-OHD levels and the probability of conception. The relative risk or odds ratio for conception is on the y-axis, while the first author is on the x-axis. And all 4 studies report an increased relative risk of conception for higher levels of 25-OHD, although the level of precision varies and the level of 25-OHD examined also varies across the studies. And this is shown below the first author name. You can see what the cutoff was, or that in the one study, they divided it into tertiles. So regardless of the exact cut point for 25-OHD, the studies consistently show an elevated probability of pregnancy with increased 25-OHD. So in summary, the literature supports an association between vitamin D and conception. More studies are needed to increase sample size and precision for the estimated associations. In our study, the association was seen at high levels of 25-OHD. So the answer to our second research question was also yes, but we needed more data, and we feel we need larger studies to precisely estimate the strength of the association or the dose response association between 25-OHD and probability of conception. So our next research question was whether vitamin D is related to early crown rump length in the first trimester. But before we could examine vitamin D, we needed to identify all the factors that were associated with crown rump length as potential adjustment factors in our analysis. And a graduate student in my group is working on this really novel analysis that uses ovulation-based gestational age to account for differences in crown rump length due to age. And so far, these are a couple of her preliminary results. She has found that older females have fetuses that were 16% longer, while older males had fetuses that were 1% shorter per year of male age. Parity was also related to first trimester crown rump length, with those with one previous birth had a 5% shorter crown rump length, and those with 2 or more previous births had fetuses that were 17% shorter. So the analysis of first trimester crown rump length is currently in process, and we hope to have vitamin D results soon, but I couldn't resist showing you some of her novel results. The use of ovulation-based gestational age is something really new in this literature, and I was excited with her findings. So then our next question was whether vitamin D was associated with pregnancy loss in our data. And in this study, exposure was, again, vitamin D, measured as 25-OHD. And here we defined low vitamin D as less than 30 nanograms per mil. Remember that our sample size would be decreasing. So we have women at preconception, they have their vitamin D measured, but only a subset of them will conceive. And so the sample size becomes smaller, and women who have higher vitamin D are more likely to conceive. So the distribution is also less deficiency. So we looked at time to miscarriage as the outcome, where miscarriage was a pregnancy loss before 20 weeks of gestation, and then gestational age at the time of the positive pregnancy test and at the time of miscarriage was defined by ovulation. So similar to the other study I told you about with crown rump length. Time to miscarriage in days was modeled with a multivariate adjusted Cox proportional hazards model. Women were at risk of miscarriage from the day of the positive pregnancy test until a loss. And then we used multiple imputation to impute day of ovulation where it was missing. Remember that we had not distributed the ovulation test kits until later in the study's lifetime. And so we have a significant proportion of people missing an ovulation day for their conception. 25-OHD wasn't associated with lower risk of miscarriage in our analysis. In both the unadjusted and the adjusted analyses, we don't see any protective effect of 25-OHD. And I'm not showing the continuous results, but they are similar. So we did not find an association between vitamin D and pregnancy loss in our data. We do see associations between menstrual cycle length, probability of conception, and vitamin D. So we began to ask about potential biological pathways that might be involved in our associations. The first pathway we considered was through ovarian reserve. We thought that vitamin D may influence ovarian reserve, which might affect menstrual cycle length or ovulation timing. And we first examined this in the uterine fibroid study, which was conducted through a health plan in Washington, D.C. And this population was age 30 to 49. Almost 60% of participants were Black, 1/3 were obese, and 3/4 had a 25-OHD level below 20 nanograms per mil. And here we found that increasing 25-OHD was associated with decreasing FSH. Increasing FSH indicates a lower ovarian reserve. And this was stronger for younger women and stronger actually for participants of Black race. So we followed up on these results with data from the time to conceive cohort. And we've already discussed the measurement of 25-OHD. We measured AMH, FSH, and inhibin B. And we used linear regression and logistic regression to look at both continuous hormonal endpoints and dichotomous hormonal endpoints. And here, these are the results from that time to conceive analysis. We looked at 4 different dimensions of 25-OHD exposure, and we saw that vitamin D of less than 30 nanograms per mil was associated with increased odds of low AMH, but not increased odds of high FSH. So those are the circled results. This is insufficient with low AMH, and here's the FSH, and there's no association with high FSH as a dichotomous variable. So in fact, the continuous measure of 25-OHD was associated with reduced odds of high AMH. So insufficiency was associated with low AMH, which is decreased ovarian reserve, but not high FSH, which also would indicate decreased ovarian reserve. So we would expect those two to go in the same direction, and they don't. And in fact, this one tends to go, while it's not significant, it tends to go in the opposite direction. So our ovarian reserve results were inconsistent. We had few women that had insufficiency, vitamin D insufficiency, and low AMH and not high AMH. We had few women that had both low AMH and insufficient 25-OHD. And we don't see strong evidence of an association between 25-OHD and AMH or FSH in our time to conceive data. And then recently there was this meta-analysis, systematic review and meta-analysis published on this topic. And there were 18 observational studies and 6 interventional studies, and they only meta-analyzed the trial results. But you can see the results here for both non-PCOS populations and PCOS populations, but they seem to not agree or go in the same direction. So the observational studies that were not meta-analyzed are also inconsistent. And so in my view, this is an unresolved research question. There may be an association between 25-OHD and ovarian reserve biomarkers, and it may be different for women who have PCOS versus non-PCOS populations. So another pathway we thought might be related to vitamin D was chronic inflammation. there are data from women with polycystic ovary syndrome and endometriosis that suggest inflammation may contribute to ovulatory disorders or subfertility, even independently of obesity. And C-reactive protein is a known marker of systemic inflammation. CDC and the American Heart Association recommend it to identify people at higher risk of cardiovascular events. And among apparently healthy women, CRP predicts a 5 to 7-fold increase in the risk of vascular events, like approximately 8 years later. So for example, So our hypothesis was that chronic inflammation might lead to reproductive disturbances and that vitamin D might intervene to reduce that effect. We investigated this hypothesis in our time to conceive study population where we measured C-reactive protein using a high sensitivity immunoturbidimetric assay at Duke University. And then we examined whether it was related to fecundability using those discrete time models I described previously. And overall, CRP was not strongly associated with fecundability. There's potentially decreased fecundability at the high level of CRP, but it was not conclusive and the sample size was pretty small. And in this figure, I've added a stratification or interaction between CRP and 25-OHD level. And the null association between CRP and fecundability was maintained across the vitamin D categories with one exception. Women with high 25-OHD level and high CRP, this estimate right here, had improved fecundability. They seemed to conceive faster. This was counterintuitive to our a priori hypothesis regarding inflammation and reproductive function. So in total, CRP was not clearly associated with fecundability overall. High levels of CRP were imprecisely associated with reduced fecundability, but we had a small number of participants with this high level of CRP. However, I do think it indicates that high levels of CRP are often excluded from analyses as a marker of acute illness. And I think this suggests that there may be systematic associations with high levels of CRP and that they may be worth including in future studies to see if they're associated with health outcomes. High CRP was associated with higher fecundability only in this one level of 25-OHD of greater than 40 nanograms per mil. There are small n's in that category. There's no dose response. And so we essentially considered this a spurious association. And we conclude either way that CRP does not underlie the associations we saw before of 25-OHD with higher fecundability. So the biological pathways linking vitamin D with either menstrual cycles or fertility are still unknown. Most of the animal studies that I referred to in the introduction didn't propose a mechanism for the observed associations. Some of the animal studies suggest decreased estrogen synthesis. However, few human studies have examined estrogen and vitamin D associations. Lower vitamin D was associated with lower estrogen in 2 human studies, one of which was a study I collaborated on with a colleague in my branch and at NICHD. And this figure shows those results, that 25-OHD less than 30 nanograms per mil is associated with lower estradiol across the menstrual cycle in healthy cycling women. However, in 2 other studies, one did not find an association and one reported an inverse association. There's been one clinical trial of vitamin D that found lower progesterone on day 21 of the menstrual cycle, but they did not account for any changes in ovulation timing, which might be influenced by vitamin D. So from this, I concluded that more data are needed to characterize the endocrine response to vitamin D treatment. And so, not sure what that is. And so I proposed the investigation of vitamin D and menstrual cycles trial, or INVITED. To better understand the effects of vitamin D on reproductive function, I've launched the INVITED trial. And the primary aim of the trial is to examine the effect of vitamin D supplementation on hypothalamic pituitary ovarian axis hormones. And I'll describe this in more detail in just a couple slides. There are several secondary aims for the trial, including examining the effects of vitamin D supplementation on endometrial decidualization, which I will again say more about later. Also to quantify the increase in 25-OHD after 1.5 and 3 months of supplementation. There are actually few previous studies that have examined the acute increase in 25-OHD with supplementation. And for reproductive endpoints, it may be relevant to know how quickly 25-OHD increases with supplementation and how quickly the menstrual cycle changes are observed. So I think this is an important gap in the literature. We'll also look at the effect of vitamin D supplementation on the circulating metabolome. This aim will allow the identification of novel pathways of vitamin D action that could be targeted in future studies. And this aim is performed in conjunction with our studies of vitamin D and reproduction in the mouse. And so we'll actually be able to compare metabolomic changes in the human samples with the changes that occur in our mouse model. So the Invited trial is a collaboration with the Henry Ford Health System in Detroit, Michigan, and that's the primary study site. It was also the primary study site for SELF, and we were recruiting using traditional and mobile app-based recruitment. By traditional, I mean social media, electronic medical record data, community flyers, and for app-based recruitment, we're partnering with Ovia Fertility and Clue, which are mobile apps used for menstrual cycle tracking. These companies will be able to push an advertisement for our study to their users in Michigan. And once women see the ad, they can then visit our website and join the study. For the study, our eligibility criteria include menstrual cycle length of less than 50 days, female aged 19 to 40 years. If sexually active, they have to be using a non-hormonal contraceptive method, and they can't be using vitamin D or calcium supplements, and a BMI of less than or equal to 35. So the vitamin D trial has 2 phases, a pre-supplementation phase, that's 1 menstrual cycle long. Women collect daily urine specimens and daily diaries. And then we measure their 25-OHD, and depending on their level of 25-OHD, they're invited into phase 2 where they're supplemented with vitamin D3, or they receive a placebo if their 25-OHD level was sufficient. And in the second phase of the study, they provide 2 cycles. They complete ovulation tests. And in the third cycle of the second phase, they again collect daily urine specimens and daily diaries. So for the primary aim of the trial, we're going to compare the following hormonal markers, both pre and post supplementation. The first is mid-luteal progesterone, which is the most consistent and well-established sign of a healthy and fertile follicle. We're also going to look at the rate of rise in follicular phase estrogen as an indicator of follicle development. Prolonged cycles may result from reduced estrogen production, and healthy follicle development could be indicated by a consistent rise in preovulatory estrogen. We're also going to look at preovulatory LH as an indicator of healthy ovarian hypothalamic interaction. And all of these hormonal markers will be measured by Nanette Santoro at the University of Colorado using funds from a Bench to Bedside award that I received about 1.5 years ago. So I want to say a little bit more about our secondary aim to examine decidualization using menstrual blood collected in the Invited trial. First, I just want to explain that decidualization is the process of the uterus remodeling itself to prepare for pregnancy. In particular, uterine stromal cells shown here change their shape and begin to secrete numerous proteins and cytokines that influence the surrounding tissue and ultimately facilitate implantation. And one protein that's secreted is insulin-like growth factor binding protein 1, or IGF-BP1, which can be used as a marker of decidualization. So to examine decidualization, we're going to collect menstrual blood from approximately 40 women. They will collect in 2 menstrual cycles, one before and one after vitamin D supplementation. And we're using a published menstrual blood collection protocol that includes Diva cups. For anyone unfamiliar with the Diva cup, it's shown in the picture here, and it's a small, flexible, bell-shaped silicone cup that is inserted into the vagina to collect period flow rather than absorb it like a tampon or a pad would. And these Diva cups will be provided to us for free. From the menstrual blood, we will isolate menstrual serum and stromal cells. Menstrual serum is what we call the supernatant after menstrual blood is centrifuged. So we'll have two ways of investigating the effects of vitamin D on the uterus. We recently performed a pilot study to examine whether vitamin D is detectable in menstrual serum, and in the 3 samples we tested, it was detected. So we're excited to follow up on these results in the main trial, where we can use menstrual serum as an indicator of how 25-hydroxy-D changes with before and after supplementation. However, the primary endpoint for the menstrual blood analysis is stromal cell function, and we'll examine decidualization capacity and progesterone sensitivity before and after supplementation. And so we actually did a pilot study of this with stored human uterine stromal cells that were isolated from menstrual blood. And we stimulated these cells to decidualize both with and without progesterone. And decidualization was measured with IGF-BP1. Remember that higher IGF-BP1 indicates greater decidualization. And the figure shows that in control cells not treated with vitamin D, treatment with progesterone slightly increases the decidualization response. So we can stimulate the cells to decidualize, and adding progesterone, or in the absence of progesterone, and they will decidualize. And then when treated with progesterone, you can see that the decidualization response slightly increases. These are all in control cells. Vitamin D treatment led to a stronger decidualization response, even in the absence of progesterone. So the 2 blue bars show in the control cells, the decidualization response in the absence of progesterone, and then the higher decidualization response in the absence of progesterone, but with treatment with vitamin D prior to the decidualization stimulus. However, the decidualization response was even stronger with progesterone. So now you're comparing the 2 orange bars. And again, you see that the decidualization response is much higher when the cells were treated with vitamin D prior to the decidualization stimulation. So it's possible that vitamin D enhances the effects of progesterone on decidualization, and we're looking forward to repeating these experiments with the menstrual blood samples we're collecting in Invited. And I think this is a good place to point out the connection between those in vitro decidualization results and our investigations in the mouse. And I'm going to really quickly go through this. So I won't explain the whole project. I just want to show you some results that are in agreement with the in vitro study I just showed you. So in this study, mice were fed one of 2 diets, one that had vitamin D and one that was deficient. And then the mice underwent an artificial decidualization procedure. And then we compared the uteri between the vitamin D deficient and the vitamin D replete diet. And we found that the mice that were given a vitamin D deficient diet, decidualization was less robust. So the figure shows that the uteri from vitamin D replete mice in the left column were larger, indicating a larger decidualization response, compared with the right column, which shows a relatively small decidualization response in the vitamin D deficient mice. And to look at that quantitatively, the decidualization response was about half as strong in the mice fed the vitamin D deficient diet. So to summarize the trial, it synthesizes both animal and human observational studies into public health relevant translational research. The trial investigates vitamin D's influence on the hypothalamic pituitary ovarian axis by careful evaluation of hormonal changes that occur with vitamin D treatment. And I just want to acknowledge very quickly my collaborators on this trial, and also the fellows in my lab and all the other collaborators for all the other results I presented. I really appreciate their support. And one plug for working at NIEHS, I'm currently hiring for a postdoctoral fellow. And there are several investigators that are hiring in our branch. If you scan the QR code, it'll take you to our the link to the job announcement. So please feel free to look at that or pass it on to friends you know are looking for a position. And I'm happy to take questions. Thank you for having me. I appreciate it.
Thank you very much, Dr. Yukish. This is a very comprehensive and amazing talk about vitamin D and reproductive function, not only to menstrual cycles, but to nearly every perspectives in a woman's reproductive event. So I think we have some time for one or two questions. So if you have any comments or thoughts to share or questions, please turn on your video and speak up. I think Shruti has a question. Go ahead, Shruti.
Thank you so much. This was such a lovely, comprehensive talk, and you took us through from observational studies to the potential for translation. That was exciting. So I just had a few questions, and if anyone else has a question during this time, just interrupt me. I took down a few questions in case there weren't any questions, but let's just start. So initially, you presented one study around vitamin D and ovarian reserve looking at AMH and FSH. And one question, because a lot of times we think about the ratio of estradiol to FSH in understanding ovarian reserve. And I saw you focused on FSH only as your main value to interpret ovarian reserve. How did you think about using the relationship between FSH and estradiol, because sometimes a diminished reserve can be unmasked by an elevated estradiol, even in the setting of a normal or subnormal FSH.
Oh, that's interesting. So, well, you'll have to tell me more about that because we didn't consider estradiol. We do have a measure of it, but just at that one time point.
What time point was that?
That's early on the day 1, 2, or 3 of the cycle.
Okay. Yeah, definitely we can close the loop offline and our next time we talk, but just physiologically speaking, the ratio between FSH and estradiol in the basal phase of the menstrual cycle for my team is how we understand whether the ovaries are responding appropriately in response to an FSH signal from the anterior pituitary, or if there is a little bit of a diminished reserve scenario, there is an actual elevated estradiol response, which then can feedback inhibit FSH, giving you a false normal. But we can talk more about this. Maybe if you include estradiol, you'll actually see that your AMH and FSH to estradiol ratios are congruent.
Right, that might account for. So there could be some essentially like confounding by or misclassification.
I think so. Yeah, maybe more misclassification potentially. Yeah. All right. So my other question was very interesting about vitamin D and cycle length. Clinically speaking, I do check and replete vitamin Ds for my patients with menstrual irregularity. I was wondering if in your look at the data in your own studies, are there elements of your observational either studies or reviews that support causality?
That's a big question. It's all, I'm an epidemiologist. So my bias is always that observational studies can be causal. And so, it's prospective. So measuring vitamin D prior to the onset of, we could go through, I guess, all the causal criteria. So, looking at, yes, it's prospective. People don't know that their cycles are long or don't know what their vitamin D level is. So you have also that element of blinding of the participants. They don't know. They're just reporting their cycles prospectively. They don't know when they ovulate, and they don't know how that relates to their vitamin D either, So other than that, the cohesiveness of the data to me is the best story for causality. The fact that you can see it across populations, like our cross-sectional studies seem to line up with our prospective studies. For conception, we looked at all these other studies in the literature. Everyone seems to show this increase in conception probability. So that to me, that this picture of all the literature and all the endpoints we've looked at coming together to support the idea is the best evidence for causality. And now we started the trial, so I'll let you know how that goes.
Wonderful. Okay, last question. I've maybe 2 more questions if no one else chimes in. Next question is, what do you think the limitations of using time to pregnancy studies are for understanding exposure outcome relationships with menstrual cycle length? If any, maybe.
So if we're, if we're looking at menstrual cycle length, you mean, and then using a time to pregnancy. Yeah, I mean, the biggest limitation is the one we tried to account for. So we used inverse probability weighting to account for the fact that some people have more cycles than others. And so if people conceive quickly, then you only get one cycle from that person. And you get 3 or 4 or 5 cycles from somebody who conceives less quickly, potentially. And so there's some potential bias there. And we did try to account for that by weighting. Honestly, it's the other problem, especially for the trial, is we're trying to look at cycles over time. And so you can't really look at people trying to become pregnant. They fall out of your study. So if I'm trying to say, oh, I gave you, you're vitamin D deficient, and now I'm going to give you vitamin D, and now I want to see what happens to your menstrual cycles. If people are trying to become pregnant, again, they're falling out of your study before you can really identify whether their cycles have changed. So it's a little bit tricky trying to look at menstrual cycles in a time to pregnancy population. But I do think that it works. I don't, like I said, if you try to account for the bias, I think that you get a good answer. But there isn't a whole lot of options for menstrual cycle research.
True. And well stated. One of the other thoughts I have for those entering into time to conception studies, if there is some sort of enrollment criteria of, 0 to 3 months of trying, I think those with irregular cycles or longer cycle lengths have already met that criteria and are no longer eligible. So something I think about. And last question. So vitamin D does, the hormone does rely, at least on the skin conversion, on cholesterol. And then you mentioned progesterone, which is another cholesterol steroid hormone. And there are known fluctuations in kind of cholesterol over the cycle based on functionality of the corpus luteum. And this is more of kind of a reach question. I don't expect you to have an answer, but I'd love your thoughts. Do you think that there is a potential influence of kind of that substrate, which is circulating cholesterol, on kind of peak concentrations of vitamin D or progesterone, and how could that influence your interpretations of kind of vitamin D in the luteal phase based on the biosynthesis?
Yeah, so if I understand the question right, it's how cholesterol levels might influence your ability to produce vitamin D, right? Is that so, and honestly, I'd like to hear your opinion on this, but what I would say is it seems that that doesn't seem to be much of a limiting factor in terms of vitamin D. What we do have is, most people seem to be replete in cholesterol. We don't have cholesterol deficiencies, in the US at least. That doesn't seem to be an issue. So that wouldn't be like a rate limiting factor. And what we do see is actually the opposite, sort of, in that obesity makes you have less vitamin D. So people with obesity or overweight tend to have lower vitamin D levels. So I guess I don't know the correlation between cholesterol levels and how that might impact your vitamin D, but if anything, it seems to be maybe counter to that pathway of synthesis.
Thank you so much. So interesting. And I know I'll turn it back over to Hui Chu for concluding remarks.
Thank you. Thank you very much for this very nice discussion. And since we are 3 minutes out of time, so I will close this session. So thank you again for your time to join us, Dr. Yu. And also thank you for everyone who can make it to this lecture. So if you're interested in our lecture series in the future, please join us and stay tuned for our advertisements in our emails. So, and also please don't forget to fill out the survey, which has been sent